Here's the Unit 1.2 PBS review. So the first essential question is, what is DNA? And we talked all about the structure of DNA as you built the nodules. The DNA stands for deoxyribose nucleic acid. It's a nucleic acid polymer. And it means polymers means it's made of many monomers. We're going to talk about that later. And in those monomers, it's made of a deoxyribose sugar opposed to other sugars, which we'll again talk about later. The shape of DNA is a double helix. It has two strands, and it's a twisted ladder that looks like this. And it's made of many repeating nucleotides. So here would be one, here would be one, here would be one, and so on and so forth, all the way up the ladder. Within that ladder, it's made up of a sugar phosphate backbone that goes all the way both sides, all the way down both sides. Both of these little bluish gray pieces are the sugar phosphate backbone. And then in the middle is always two bases that are connected. A and T, adenine and thymine, are always connected. Guanine and cytosine are always connected. A little bit more about the bases. These bases are pyrimidines, thymine and cytosine. You can remember that because they have Ys. They only have one ring, whereas the nitrogenous bases, adenine and guanine, are purines, and they both have two rings. And again, cytosine and guanine pair together, and between them there's actually 3 hydrogen bonds. And adenine and thymine always pair together, and between them there's always 2 hydrogen bonds. The next question was, how do scientists isolate DNA in order to study it? And basically they go through a 3-step process. If using plants, the food needs to be mashed up first. If you're using human spit, you can spit into a cup. You mix it with a soapy/salty solution that's added to, and that breaks up the cell membrane, and so frees the DNA from the cell. And then you can add alcohol, which will separate the DNA from the rest of the liquid. If you want more information on this process, you can go to this website. I'm not going to take you through it because it's just the simple basic process that we did in class with the lab. But you can review it if you go to that website. Moving on, how does DNA differ from person to person? So this is where we used restriction. Even before that, just DNA differences from person to person based on the bases. So if you would take 2 pieces of DNA from 2 different people, most likely, depending on where you take it from, there would be a slight difference in base pairing, and you can see that in this gene here. But there's only about a 0.1% difference on average between humans. Alright, so every so often there's a random different base pairing. Now, we can use molecular biology to identify people by using restriction enzymes, which make restriction fragment length polymorphisms, RFLPs, and then we can also use gel electrophoresis. So we'll go through that, review that quick process here. So restriction enzymes are used to cut the DNA into different sizes of RFLPs that are determined by the sequence. So restriction enzymes only cut at a specific sequence, and that sequence could be in different spots in the DNA depending on the person. And that will create different size fragments. The DNA is then run on a gel to separate the fragments, which we'll talk about in a little bit. And then the fragment lengths can be compared on gel using the bands on the gel. To review this process, there's a really good video. I'm actually going to pull it up now. So just to review the process. So here we have 2 different pieces of DNA, and as you can see, well, that's actually 1 strand of DNA. And as they go through, they're going to show the restriction sites here. And now here's a different strand from individual B. So you notice how these 2 sites here on the either ends are the same, but the site in the middle is different. So the restriction enzyme only cuts at this specific site, GAATTC. So an individual A is going to show in a second will get cut 1, 2, 3 times. So therefore they would eventually have these 2 segments, plus they would have segments on either side if there was continued DNA. Whereas the individual B didn't get cut in the middle, so it only made one larger fragment here, and there would be other ones here. And then if we put it on the gel, which they're going to show you here, remember the shorter fragments go the farthest on the gel, where the longest stay back. So individual A would actually have 2 fragments, and one would be short and one would be slightly longer, and then the other individual B would have just one long fragment because that's all the more it was cut. So going back, and we can continue to review gel electrophoresis here. So DNA is run on a gel and is propelled by a current, and DNA is negatively charged, so it's always going to go to the positive end. So the positive end of this gel would be down here, the negative end would be up here. If you switched it around, the DNA would all run this way and probably off your gel, and you wouldn't see anything. The smaller magnet fragments move a lot farther in that time because they can fit through the gel a lot easier, whereas the longer fragments take a lot longer to move, so they don't move as far in the same amount of time. So the longer bands are up here, the shorter bands are down here. And then what we can do is we can take DNA found at the scene and we can compare it to our different fragments. And using this picture here, it would have been dino because these fragments are the exact same, so that means he is probably his sample is probably the one that we found at the crime scene. And that is it for this unit. So you just need to know the structure of DNA, restriction enzymes, and how gel electrophoresis works.